Good afternoon, and thank you to everyone joining our talk in person and online. This talk is part of our Seminar for Non-Scientific Staff series, which allows support staff and external people to gain insight into the research undertaken by our lab members. Before we begin, there's just some technical details. At the end of this talk, there'll be an opportunity to ask questions. If you're participating in the seminar online, please write your questions in the Q&A box on Zoom, and we'll read them out during the question session at the end. The MRC Laboratory of Molecular Biology, or LMB, is a world-leading research institute dedicated to understanding important biological processes at the levels of atoms, molecules, cells, and organisms. We aim to study difficult long-term problems, and in doing so, to contribute knowledge needed to solve key issues in human health. We seek to understand the structures of molecules and molecular machines, their fates and functions within cells, and how these make up complex systems such as tissues, organs, and the immune system and the brain. The LMB's origins date back to 1947, when a unit was funded to study protein structure. Over the years, LMB scientists have made many revolutionary contributions to medicine, both in terms of new understanding and in the development of new technology. Our talk today is given by Patrycja Kozik. Patrycja joined the LMB in 2016 and became a group leader in the protein nucleic acid chemistry division. She did her undergraduate degree in Germany at the Jacobs University in Bremen. Following her PhD in the lab of Professor Margaret Scotty Robinson at the Cambridge Institute of Biomedical Research, Patrycja was awarded a Sir Henry Wellcome postdoctoral fellowship for a collaborative project between the Institut Curie in Paris and the Broad Institute of Harvard and MIT in Boston. In her current research, Patrycja combines her interest in protein trafficking and immunology to study how the unique cell biological properties of dendritic cells enable them to effectively control infections and cancers. I'll now hand over to Patrycja, who will explain her work and research in more detail.
Thank you very much, Laura. Great. So, welcome everyone. So this is the title of my talk, and this is my lab. Some of them are actually sitting there at the back, so I think they will veto if they disagree with anything I'm saying. So I have to be more careful. I wasn't expecting that. So this is us with them actually on top of a hill at the Peak District, and normally we're on level 3 here at the LMB. So the question we'd like to address in the lab is how to identify and eliminate cancer cells. So I'm going to talk about this today, and also how the immune responses against cancer cells are similar to the immune responses against viruses, and how all of that is similar to finding Wally. So how do we identify and eliminate cancer cells? So in the modern society, if you have some individuals that you'd like to eliminate because they cause disruption, normally we would use police forces to detain such persons and isolate them. In the immune system, infected and cancer cells are eliminated by so-called T cells or T lymphocytes. So T lymphocytes are very interesting cells because they can efficiently go and kill the cells that are either infected or cancers. So here we have T cells in green and an infected cell in red. And you can see is that these cells sort of crawl around, and when the T cell comes into contact with a cancer cell, the cell suddenly lights up. So this lighting up means the cell died because the membrane became leaky and it incorporated all of this red dye, and then it disintegrates into small bits. So how do T cells kill the targets? So if we look closely at this interaction between the T cell and the target cell, what happens there is that T cells secrete a lot of toxic proteins into this narrow gap between the cells. So we can see that here. One thing that the T cells secrete is this blue thing, which is a pore-forming protein that makes holes in the membrane. These holes partially destabilize the cells because the integrity of the membrane surrounding the cell is compromised, but also they allow for these red things here, the different toxic proteins, to enter, and now these can kill the cell more efficiently. So how do we recognize the target cells? How do T cells recognize their targets? So when we look for Wally, we recognize him by this white and red stripy jumper. And all cells in our body are dressed with so-called MHC class 1 molecules. So these MHC class 1 molecules, they present small fragments of proteins. So the cell is making like all the different proteins that it needs for its function. They make, then they degrade, and small fragments of these proteins are continuously presented on these MHC class 1 molecules. So that gives us sort of an overview of what's happening in the cell. When the cell is infected, in addition to these normal proteins that always should be there, there will be some proteins from a virus, because now the virus is replicating in the cell, it's making new copies of itself. So some of these viral fragments will also end up now in the MHC class 1 molecules. So these are recognized as so-called foreign peptides. And similarly during cancer, because cancer cells accumulate a lot of mutations, so they have a lot of proteins that are slightly wonky and look a little bit different. And these different peptide fragments can also be presented on MHC class 1 molecules and also recognized by T cells as foreign. So T cells use a T cell receptor, a receptor that will now recognize MHC class 1 with foreign, not with self-peptides, and then a signaling from that receptor sends a signal to T cell to now secrete all the cytotoxic molecules and kill. So you might have not heard about the MHC class 1 molecules, but you have probably all heard about donor matching. So how is donor matching relevant here? What are we actually matching? So one of the things you need to match between the donor and the recipient is actually these MHC class 1 molecules, because we all have slightly different MHC class 1 molecules. So if you put a tissue from someone with different MHC, now actually it doesn't matter anymore what peptide it presents, because that's recognized by a T cell as foreign anyway. So that's why you can get rejection of a transplanted organ, because it's basically killed by the T cells recognized as either cancer or an infected cell. So each T cell receptor recognizes a specific combination of an MHC class 1 molecule with a peptide. And we have about 1 million to 100 million different TCRs in the body. So each T cell will express only one T cell receptor. So each T cell can recognize only a specific cell infected with a virus or a specific cancer cell. And we have about 40 billion of T cells overall, which corresponds to about 3 million in a teaspoon of human blood. So this is quite a lot of T cells that could potentially cause a lot of damage if their activity were uncontrolled. And it's really important to make sure that T cells don't accidentally kill healthy cells. And there is a whole range of different mechanisms to ensure that T cells don't go out of control, but it sometimes does happen in diseases such as celiac disease or type 2 diabetes. One of the ways in which we make sure that T cells only kill when it's needed is that the T cells in the blood, they circulate in so-called naive state. That means that they're very small, not very active. They actually don't produce any of these cytotoxic molecules, so they cannot kill. Even if they were to recognize MHC class 1 presenting a peptide, they actually can't do anything. So before the immune response can start, we actually need to activate the T cells that have the right TCR for a given pathogen or for a given mutated protein. So how do we decide which cells to kill? So how did we decide to look for Wally? Well, I actually don't know. This is probably going to ask the author of the cartoon, Martin Hanford. But for the purposes of this talk, we're going to pretend that this decision was made by the head office. So the head officers of the immune system are our favorite cells, or it's my favorite cells, dendritic cells. Dendritic cells survey all the tissues. So you can see them here. They have these very long projections, and they use them to sort of sample the environment. So they will be present in most of the tissues in the body, and especially those tissues which are exposed to extracellular environments, so skin, lungs, intestine. So cells which are more likely to be infected or are exposed to things such as UV radiation, so they're more likely to undergo mutations, and therefore you need to start this immune responses quite quickly. So we have dendritic cells sampling a tissue, and when it recognizes some sort of danger signals, it then migrates into the lymph node, so the closest lymph node that's the proximal lymph node to that tissue. And in that lymph node, it now presents the same MHC class 1 peptide complexes as the the cancer cell, so the target cell, so that it can now find the T lymphocytes that will have the relevant TCR. So what happens? The T lymphocytes, they circulate through the blood and they kind of come into all the lymph nodes while they do it. And if they encounter a dendritic cell presenting the peptide that they can recognize, then they stop and they start proliferating. So you make loads of copies of the same T cell with the same TCR, so you can build up the army. They start expressing the cytotoxic molecules. And this process is one of the reasons why during infections the lymph nodes swell, because you're sending a lot of signals to recruit new T cells, and these T cells really need to proliferate and expand multiple times. And you can see here just a diagram showing that the lymph nodes are sort of distributed throughout the whole body, and some of them you would have probably had better, those sort of in the neck, right? Because when you have a cold, these are more likely to swell, or these armpits that are often tested for metastasis of certain cancer. So these cells now that proliferated and became activated go back into the blood, and through the blood, they can go into the target tissue and do the killing. So this sounds, well, I was going to say straightforward. It probably doesn't sound straightforward, but it's even more complicated than that. So the question is now, so as I mentioned, these peptides presented by MHC class 1 molecules, they're sort of made in the cell while the cell is going about its normal business. But now the dendritic cell has to present the same peptides with the same MHC. So how does it do it if it doesn't actually make those proteins, right? Because the virus maybe infected only the cell and the tissue. It didn't actually infect the dendritic cell. Similarly, if it's an immune response against the cancer cell will have all the mutations, but the dendritic cell does not actually have those proteins with the mutations. So this is one of the things we study in the lab. So these peptides are made by these sort of big barrel proteins, which are called proteasomes. So the proteins kind of go in and they chopped up inside and then come out as these short peptides. And we know that in dendritic cells, the same thing has to happen. They somehow have to be chopped up into the small peptide now in the dendritic cell. So we've been interested in how these proteins from the cancer cell or a virally infected cell actually make it all the way into the dendritic cell, and then where they can be chopped up and then later presented on MHC class 1. One of the things that was particularly interested to me, because I'm sort of generally interested how cells have different compartments inside, and these compartments allow them to sort of keep different processes separated from each other. So you have these different bubbles, and like one bubble maybe will be involved in degrading proteins, another one will be involved in making new proteins, and another one might be involved in sort of modifying them or the cell respiration. So one dendritic cell, so this is now a dendritic cell, and it's taking up here bacteria actually rather than cell fragments, but cell fragments would be approximately a similar size. So what you can see is that when it takes up these cell fragments, they end up in another vesicle inside the cell. Oops. Right. But this barrel degrading machine is actually here. So somehow they have to get out of this little vesicle and make it into the cytosol. So we wanted to find the mechanism of how this happens. So to do that, we decided to use a toxin. So it's a plant toxin called saporin, and it's very similar to another protein called ricin, which you might have heard about, because ricin is actually an extremely potent toxin. So the reason why ricin is extremely potent is because ricin has a way to get into the cells very efficiently. Saporin doesn't actually get into the cells very efficiently, but once it is inside the cell, it is equally efficient as ricin. So what makes the difference between the saporin and ricin is that saporin actually cannot get to the target very well. But we could use it as a way to actually study how dendritic cells leak these proteins into the cytosol. So to do that, we made a so-called library. So we took a population of dendritic cells, and in each dendritic cell, we knocked out a specific gene. So now each of these Cs has a gene missing. So some of the functions in these cells will be disrupted. We then added saporin, And in most of the cells, we were expecting sporin now to escape into the cytosol and be toxic. And the question was, do any of those cells where we disrupted something will accumulate sporin in these vesicles and will never let sporin inside into the cell? So we found some cells where that was indeed happening, and then we could see what protein is actually missing here. And what was quite exciting for us is that the protein missing was also a pore-forming protein, so very similar to this blue pore-forming protein made by T cells. So that suggested that T cells make a pore-forming protein secreted that makes holes in the target cell. But before that can happen, dendritic cells use also a pore-forming protein that they use in order to deliver things from outside of the cell into the inside of the cell. But that is not toxic, that is used to actually facilitate the immune response. So different immune cells use different of these different pore-forming proteins to actually for an immune response to be efficient. So what we are now trying to understand in the lab is how is it that they can actually control the activity of this protein? Are you making a protein that can make holes in your own membranes? How does it not go out of control and just kill the cell altogether? So this is sorry, I got stuck. Maybe I'll just have some water to distract myself. Right, so this is one of the questions we're trying to address. But the second question is, so, this is how the initiation of the immune response by dendritic cell takes place. So they take up fragments of the cells, present them on their MHC class 1, deliver them into the cytosol, present them on the MHC class 1, T cell gets activated, and then it can kill the cell expressing the same MHC on the surface. But how does the dendritic cell know that this is actually the cell we want to kill, right? So how do dendritic cells identify the cells that need to be killed in the first place? So this is the second question we're trying to address in the lab. So if Wally was a terrorist, one of the very clear indicators that he needs to be isolated is if he was carrying a bomb. And when cells are infected with a virus or a bacteria, it is also much clearer that these cells need to be eliminated. Bacteria, viruses, and all pathogens have a lot of molecules that are not present in mammalian cells. So this could be different like sugars, different lipids, or just different molecules that mammalian cells do not have altogether. So if the immune system detects those molecules, it's pretty clear that the cell is infected and needs to be eliminated. But cancer cells are actually quite similar to all the other cells in the body. So it's quite unlikely that they will have something that is completely different from everything else that's present there. So the dendritic cells have to rely on much more subtle cues. One of the things could be that certain molecules will be accumulated in excessive amounts. So these could be things that are actually not toxic per se, or actually not dangerous as such. But if you have a lot of them, that's sensed by the immune system as some sort of a danger signal because something became destabilized. So if someone went and bought 1 million of matches, it will sort of raise your eyebrows. Why is that happening? Another hypothesis is that we are exploring currently is that maybe the cancer cells are actually showing the signs of stress themselves. So if you had guilty conscience and you realize you're being under surveillance, you might start acting erratically, sweating, and displaying all the signs of stress. So what we are also trying to understand is what are these signals that are produced by cancer cells are signaling to the dendritic cells that this is the cell that needs to be killed. So we do know that dendritic cells have a whole range of different receptors on the surface. So what are these receptors? So if I was a dendritic cell and like each of my arms was a different receptors, but I would have instead of 2 arms, like 100 different arms, and each arm can kind of grab something else. And I'm going blindly around and suddenly I sort of found something that, is actually fitting really well into this weird arm with a different shape. So that recognition triggers then signaling and sends a signal to the center of, to my brain, or in the case of dendritic cell, to the center of the dendritic cell. it sends a signal that this cell displays some either danger or infection signals. So some of these receptors have been very well characterized, especially those that recognize viruses, because this has been studied for a really long time. But what we try to understand now is which of these receptors might recognize the stress signals produced by cancer cells, or these molecules that might accumulate in cancer cells excessively because the cancer cell is proliferating really rapidly and trying to overcome the tissue. So just to summarize, the way the whole immune response is initiated is initially by recognizing stress signal produced by cancer cell or fragments of pathogens that are being produced during infection. This sends a signal to a dendritic cell to now start taking up things that it sees in the environment and fragments of neighboring surrounding cells. The proteins are eaten by the dendritic cell, enter the cytosol where the short peptides can be made. We now display these short peptides on the MHC class I molecules, activate T cells, and T cells go and kill the target cells. So now that we identified the danger signals, we instructed the appropriate forces within the target, we can try to find Wally. And if anyone can spot him, I was gonna bring a prize, but my slides weren't working, so instead I had to get my slides. But the Wally is here. So, so that's all well and beautiful, but usually these immune responses against cancers are not that efficient, and cancers very often actually escape the immune system and grow nevertheless. So why? In fact, there are many different ways by which the cancer cells can escape. So because they have a lot of, in general, cancer cells are genetically unstable. That means they accumulate, every time the cell divides, it gets new mutations, so it maybe acquires some new functions. It starts making some slightly different proteins that help it to sort of gain advantage, right? Because now maybe it became a bit better in using low amounts of energy that are available. So one of the things that can happen is that the cells will actually stop displaying these MHC class I molecules altogether. So they become invisible to T cells. Another possibility is that sometimes the cancer cells actually don't produce any of the stress signals. They accumulated enough mutations that they can proliferate and grow as a cancer and sort of disrupt the surrounding tissues, but they're not that different and they're not that stressed, and that they are invisible to the danger receptors present on dendritic cells. Cancer cells can also produce other signals that will actually make dendritic cells dysfunctional. So what that means, they might stop them from taking up proteins from cancer cells, or might stop them from processing them and presenting them on the MHC class I molecules. They might also provoke dendritic cells to send wrong signals to T cells. So dendritic cells are no longer able to activate the T cell and give the T cell all the appropriate signals that it needs in order to get activated and go and do the killing. Finally, sometimes the T cells that actually got activated, because they're receiving all these conflicting signals from the immune system, as I mentioned, there are lots of different ways to ensure that the T cells don't kill you. So you can actually switch them off. So what the cancer cells can also use as an evasion strategy is to switch off the T cells that already became activated, or maybe prevent them from actually entering the cancer site. So we hope that by understanding these mechanisms that dendritic cells employ in order to activate the T cells, we can actually start to use this knowledge in order to sort of use better therapeutics. And maybe just to mention, for example, one of the most common immunotherapeutics that is now being used in clinics, these checkpoint inhibitors that you might have heard about. These are molecules that help to switch the T cells back on. So when the T cells got switched, so in cancer patients where the T cells are being made, but they're not able to actually kill the cancers, these checkpoint inhibitors actually help the T cells to become active again. So with that, I'm going to end, and I'm happy to take your questions.
So if nobody has absolutely anything they want to ask or say. On that note, then, I would like to thank Patricia for an absolutely fantastic talk, and our audiences, both here and online, for joining us and for supporting the way that we can do these talks. Our next seminar for non-scientific staff will be given by Tanmay Barrett on the 22nd of September at 12 o'clock.